All right, it's time for another screencast for AP Psychology. This is the Unit 3, Part 1 screencast. In this screencast, we're going to look at the neural network and how it works to make our brain and body function. So let's get charged up for this screencast. First, let's start with phrenology. In the 1800s, phrenologists studied bumps on the skull to reveal a person's mental abilities and character traits. There were lots of phrenology societies, and people will travel to read people's goals and tell them what they're good at. Now, we don't do this anymore, but it definitely paved the way to look at the brain and its localized function. And we'll get more into that in a bit. This is a neuron or a nerve cell. There's a bushy dendrite fibers, which receive information. There's the axon fiber, which passes the message through its terminal branches to other neurons. There's the myelin sheath, which is a layer of fatty tissue that insulates them and speeds their impulses. And there's the action potential, which is a brief electrical charge that travels down its axon. If the myelin sheath degenerates, it slows communication to muscles and eventually loss of muscle control we call multiple sclerosis. Now you need to know the parts of the neuron listed here. More info on the next slides. Here we have dendrites, axon, the myelin sheath, neural impulse, and terminal branches. And here's the cell body. Now, how fast does this impulse happen? It could be anywhere from 2 to 180 miles per hour, which obviously is measured in milliseconds. Thus, your reaction to outside stimuli can take up to a quarter second or more to register. We are slower than a computer, but vastly more complex. So how does a neuron work? Neurons generate electricity from chemical events that have electrically charged ion. There is a fluid outside an axon membrane, and it's mostly positively charged. When resting, axon fluid interior is mostly negatively charged. So positively charged outside and negatively charged inside is called the resting potential. The axon surface is very selective about what it allows through its gates. We call this selectively permeable. So when an axon fires, the security parameters change. It opens its gates and positive charged ions flood through the cell membrane. This depolarizes that axon section, causing another axon channel to open, and then another, like a line of falling dominoes. During a pause in firing, called the refractory, the neuron pumps positively charged ions back outside so it can fire again. This can happen 100 or even 1,000 times a second. Most signals are excitatory, like pushing a neuron accelerator, but some are inhibitory, like pushing its brake. Action potential. This is the direction the axon terminal will move. So we have the cell body, positive rushes in, which leads to another channel that allows it to rush in while we pump the other out. And it continues down the line. Threshold is the level of stimulation required to trigger a neural impulse. When the excitatory signal exceeds an inhibitory signal by minimum intensity, the action potential occurs, and this can travel down branches into junctions with hundreds or thousands of other neurons, or with the body's muscles and glands. Now this works as an all or none response, so either you fire it or you don't. So then the question is, where does the stronger stimulation occur? It's when more neurons are fired at the same time, or more often. It doesn't change the speed or strength, but the impulse firing more neurons can lead to a stronger response. Think about pinching softly and then pinching hard. Which one hurts more? The hard fires more nerves. Now we look at where these neurons connect to the next one. This is called a synapse. This is where the meeting point between neurons occurs, and it's a small gap that is called a synaptic gap. It's less than 1/millionth of an inch wide. When an action potential reaches the knob, like terminals at the axon end, it triggers the release of chemical messengers called neurotransmitters. These neurotransmitters cross the small gap in 1/10,000th of a second and bind to receptors, much like a lock and key. Then, in the process called reuptake, the sending neuron reabsorbs the excess neurotransmitters, ready to be released again. Quick depiction on what's happening. So you have the sending neuron, the action potential, and the receiving neuron. That's the synapse. Looks like this when zoomed in. The neurotransmitters cross, bind to the receptor sites, and then the sending neurons reabsorb. Types of neurotransmitters, some of them are acetylcholine, dopamine, serotonin, norepinephrine, gamma-aminobutyric acid, known as GABA, and glutamate. More on these later this year. In this graph in your book, you need to familiarize yourself with these neurotransmitters, their function, and examples of malfunctions, as we will definitely be revisiting this later this year. So then, how do drugs and other chemicals alter neurotransmission? We have an agonist, which is a molecule that by binding to receptor sites stimulates a response. And we have an antagonist, which is a molecule that by binding to receptor sites inhibits or blocks a response. And drugs mimic these neurotransmitters. So again, quick depiction. Neurotransmitter carrying the message. Here's your binding sites. Narrowed down. We have the receptor site receiving the neuron. And then the transmitter opens the receptor site. This is an agonist. An antagonist. An antagonist mimics the neurotransmitter opening receptor site and blocks the neurotransmitter from actually doing its job. Now let's look at the nervous system as a whole. Two parts to the nervous system. The peripheral nervous system, or PNS, is responsible for gathering information, transmitting it to the rest of the body. The central nervous system, or CNS, includes the spinal cord and brain. Now the peripheral is your autonomic, controls self-regulated actions of internal organs and glands, but also somatic, or controlling voluntary movement of skeletal muscles. The central nervous system tells the peripheral what to do. Under autonomic, you have sympathetic, which is arousing, and parasympathetic, which is calming. Information travels in the nervous system through three types of neurons. There's sensory neurons, which carry messages from the body's tissue and central sectors inward to the brain. and spinal cord. There's motor neurons, which carry instructions from the central nervous system out to the muscle glands. And then there's interneurons, which are in between the two. In the peripheral nervous system, our somatic nervous system enables voluntary control over our skeletal muscles. Your somatic nervous system reports to the brain the current state of the skeletal muscles and carries instructions to your body to rise from your seat when the bell rings to get out of class. Your autonomic nervous system controls glands and the muscles of our internal organs, influencing functions as glandular activity, heartbeat, and digestion. These are done autonomously without your conscious knowledge. Then you have your sympathetic nervous system, which arouses and expands energy if something alarms you. And your sympathetic nervous system accelerates your heartbeat, raises your blood pressure, slows your digestion, and raises your blood sugar. And cool you with perspiration when you're alert and ready for action, if something were to scare you. Your parasympathetic nervous system does the opposite. It lowers heart rate, stimulates digestion, and lowers your blood sugar. So the two keep you in balance. The central nervous system is how we think, feeling, act. Listen, there's tens of billions of neurons communicating with your brain, 40 billion connected through roughly 10,000 other neurons. We end up with perhaps 400 trillion synapses, where neurons meet and greet their neighbors. They work together in a cluster network known as the neural network. The spinal cord is a two-way information highway connecting the peripheral nervous system and the brain. The neural pathways govern reflexes or are automatic responses to stimuli controlled by the inner neurons. So inputs come in to the central nervous system. They're connected to many different pathways. And then connected to even more pathways, which the brain learns by modifying certain connections in response to feedback. Which leads to output, such as playing an instrument and making beautiful music. So what are reflexes? Reflexes are controlled by your inner neurons, and they are in your spinal cord and work to stimulate reflexes. So what happens? When you touch something hot, the inner neuron, the peripheral neuron, sends a signal. Your inner neuron then interprets it and reacts before the signal ever reaches the brain. So when you touch something hot, it's why you move your finger away before you even realize it's hot. That is your inner neuron or your reflex. Next up is the endocrine system. The endocrine system is the body's slow chemical communication system. It's a set of glands that release hormones into the bloodstream. Hormones are chemical messengers that are manufactured by the endocrine glands that travel through the bloodstream and affect our tissue. Parts of the endocrine system. The most influential endocrine gland is the pituitary gland. It's a pea-sized structure located in the core of the brain, or just controlled by an adjacent area called the hypothalamus. We'll do more on that later. The pituitary releases certain hormones when it's stimulated, including growth hormones during physical development. The hypothalamus helps control the pituitary gland. thyroid gland affects metabolism. The parathyroids help regulate the level of calcium in the blood. The pancreas regulates the level of sugar in the blood. As an example of what the endocrine system can do, if you're in a scary situation or in a dangerous situation, your adrenal glands, or ANS, which is on top of the kidneys, release epinephrine and norepinephrine, which is called adrenaline. These hormones increase heart rate, blood pressure, blood sugar, providing us with a surge of energy known as the fight or flight response, which can linger a while after the danger is gone. So essentially, there's a feedback system here. The brain coordinates with the pituitary, which coordinates with other glands, which then release hormones. And the body and brain continue to communicate. So you can see that there's an intimate connection between the nerves and endocrine system. The nervous system directs endocrine secretion, which then affects the nervous system. Lesions are tiny clusters of cells in the brain that have been destroyed. Through electrical stimulation and studying lesions, we are able to electrically, chemically, or magnetically stimulate various parts of the brain and then note the effects. We can make people giggle, hear voices, turn their head, or even create feeling of falling or an out-of-body experience by simply stimulating certain parts of the brain. One scientist studying rats selectively created a lesion on the hypothalamus, which helps control hunger. The rat reduced eating to the point of starvation. Damage to other areas of the brain can produce overeating. Now, the golden age of psychological discovery is happening right now because of new technology. An electrocephalogram is an amplified recording of the tiny waves of electrical activity sweeping across the brain surface. These waves are measured by electrodes placed on the scalp. A computed tomography, or a CT scan, is a series of X-ray photographs taken from different angles and combined by computer into a composite representation of a slice of the brain structure. A positron emissions tomography, or a PET scan, is a visual display of the brain's activity that detects where a radioactive form of glucose goes while the brain performs a given task. A magnetic resonance imaging, or MRI, is a technique that uses magnetic fields and radio waves to produce computer-generated images of soft tissue. MRI scans show brain anatomy. You also have a specialized application of an MRI called a functional MRI, which can reveal the brain's functioning as well as its structure. So where the brain is especially active and where blood goes by comparing MRI scans, taken less than a second apart. Researchers can watch a specific brain area activate, showing increased oxygen, latent blood flow. So, for example, the fMRI machine detects blood rushing to the back of the brain while someone is watching images. The back of the brain is our visual cortex, but more on that in a different screencast. Because of technology and brain imaging, we have learned more about the brain in the last 30 years than in the previous 30,000. This is a brain scan of a normal person on the left and someone with schizophrenia on the right. Notice the enlarged central ventricle on the right. Schizophrenia has its origin in structural brain changes in both the white and gray matter, but more on this in a later screencast. Now, let's look at different parts of the brain. There is the older brain structures, which are the brain stem. Brain's oldest and innermost region is the brain stem. It begins where the spinal cord swells slightly after entering the skull. This slight swelling is the medulla, where it controls your heartbeat and breathing. And as some brain damaged patients in a vegetative state illustrate, we need no higher brain or conscious mind to orchestrate our heart's pumping and lungs breathing. The brainstem handles these tasks. Sitting on top of the brainstem is the thalamus. It's a pair of egg-shaped structures that act as the brain's sensory control center. The thalamus receives information from all these senses except smell and routes it to the higher brain regions that deal with seeing, hearing, tasting, and touching. It also receives some of the higher brain replies, which is then directed to the medulla or to the cerebellum. It's almost like a sensory information transmitter. The thalamus receives all this sensory information except for smell. Inside the brainstem between your ears lies the reticular formation, a neuron network that extends from the spinal cord right up through the thalamus. As a spinal cord sensory input flows to the thalamus, some of it travels through the reticular formation, which filters incoming stimuli and relays it as important to other brain areas. Your pons is a part of your brainstem. It's a structure that links your brain to your spinal cord. It handles unconscious processes and jobs such as sleep-wake cycle and breathing. It also contains several junction points for nerves that control muscles and carry information from senses to your head and face. In 1949, two scientists discovered that if you stimulate a cat's reticular formation, it almost instantly produces an awake, alert cat. Where, if you severed the reticular formation without damaging any nearby sensory pathways, the cat lapses into a coma from which it is never awakened. So the reticular formation enables arousal. The cerebellum extends from the rear of the brainstem and is the baseball-sized little brain, which is what its two wrinkled halves resemble. Cerebellum enables nonverbal learning and memory that helps us judge time, modulate emotions, and discriminate sounds and textures. That coordinates voluntary movement. Without the cerebellum, your movements would be jerky and exaggerated. Remember, the older brain functions all occur without any conscious effort. The limbic system is a neural system including the hippocampus, amygdala, and hypothalamus, located below the cerebral hemispheres, associated with emotions and drives. The hippocampus processes conscious memories, so any animal or human that has a damaged or injured hippocampus lose their ability to form new memories of facts and events. The amygdala is two lima bean-shaped neural clusters which are linked to aggression and fear. Psychologist Heinrich Kleiber and neurosurgeon Paul Bucy surgically removed a rhesus monkey's amygdala, turning the normally ill-tempered animal into the most mellow of creatures. And studies of other wild animals, including links to wolverines and wild rats, show the same effect. Remember that the brain has many different parts. Stimulating or moving one part can affect the others. The hypothalamus is a neural structure lying below the thalamus that directs several maintenance activities like eating, drinking, and body temperature, and helps govern the endocrine system via the pituitary gland. It is linked to emotion and reward. As is often the case in science, a mistake has led to awesome discoveries. When two scientists were trying to implant electrodes in a rat's reticular formation, they accidentally placed it on the hypothalamus, and they discovered that the rat continued to return to the spot where the stimulation occurred, indicating that the hypothalamus provides pleasurable rewards. They were willing to even walk across an electrified grid to stimulate the hypothalamus. These reward centers are very powerful studies. You can even, when implanted on the hypothalamus, navigate a rat through a maze by giving them pleasure for turning right or left. Experiments have also revealed the effects of dopamine. Dopamine is a related reward system. When given a dopamine increased drug or a sugar pill when looking at pictures of vacation spots, then later participants were told to imagine the different spots. The ones that got the dopamine influence seemed to prefer vacation spots associated with that dopamine-induced feeling, while the sugar pill participants showed no change in desired vacation spots. This diagram in your book is a good place to look to get each part quickly and know what they do. And I would definitely get to know these. I'll leave you with this picture I took on a cruise. It induces dopamine in my brain. That's it for now. Thanks for watching.